All right, now it's visible. Finally, it makes its appearance. So no matter how much AI wins, we can still not figure out how to connect our screens. So it's going to be fun to see in the next 2 days how our speakers struggle to put their computers on. We'll try to make sure that they don't struggle too much. But let's talk a bit about BHARAT study, what is something which we've been doing from last 1 year. Before I start giving the presentation, I must tell you this is a talk-cum-hybrid panel. We will call the longevity investigators who've been helping doing this study after my first 10 slides, which I show you. And then we can have a dialogue with them to learn and understand what are the challenges in the study, what we have learned so far, and what's the way ahead. My slides are not changing, guys. The problem still persists. Okay. So before I start, few acknowledgements. The project of this scale cannot be done without collaboration, without team. So we have a lot of investigators in the team, the Longevity India team. We have more than 20 investigators in various forms and function who are part of the team. And as a form and function, it basically means the investigators are lending their expertise domains to study what we are trying to do in Longevity India. So we have experts who will be talking after my presentation, and you'll learn why they are there also. We are also funded by ICMR, so we are an ICMR CAR team. We have a lot of people who have been hired under the CAR scheme who actually work on the ground. A lot of academic partners who are helping us to understand the data we'll be generating. And of course, as mentioned, we're getting a lot of omics help from the STEAM lab. And both Hanno and Judith here in the audience, I cannot see where. But anything to do with omics, they are the people whom we should be asking the question to. And we hope they will continue getting the support from them in the years ahead. A lot of hospitals have a partner. After all, we need clinicians to ensure the people whom we are sampling are apparently healthy and not sick. And fortunately, we are not trained to do that, and that's why we need the clinical team to do. So we have hospitals like Baptist. The Carol is here. We finished 1,000 individual sampling with Baptist right now. Ongoing sampling is going with MS Ramaya, JSS Mysore, Satya Sai Hospital, which is our ongoing partners. We have a lot of partners who give us clinical back-end support to develop blood reports, Tata 1mg, Healthians, Dr. Lal Path Lab, and Decodage. We get a lot of technical support from Brooker, and Arca AI is our AI partner who helps us develop platform for data capture. Now, of course, you need money, the green buck, which makes a lot of difference. And you can see it's not just one, it's a portally. We need a lot of money to do this kind of work. And we're always short of funds to do what we want to do. But we've been supported by ICMR through the Center for Advanced Research in Aging. Prashant Prakash has been an avid supporter since beginning. Actually, he's the one who laid the foundation in many sense for Longevity India. Pratiksha Trust, Chris Gopalakrishnan, he's actually supported our model lab. Kiran Mazumdar has supported some fellowships for our postdoctoral fellows, and many other donors who have come ahead in many different forms to support this study. And of course, we need their support continuously in many more years to come, because I don't think we will be able to solve the problem in just few years. So why we need to have this study? Well, India is aging fast. This graph you have seen in newspapers in many places before. This is not new. But what's interesting is, by 2100, 36% of Indians will actually be in the senior citizen category. They will get privileged seats on the buses. They'll get fare discounts. They will be standing in the lines wherever they want. But the point is the number is going to be very massive, very large. And we will have a smaller number of people taking care of them. Now, this is a big worry for India. Because India, we always take pride as we are the youngest country. And think about when all these young people are going to get old together, who's going to take care of them? So a lot of people have asked me, is longevity really a problem for India right now? Is aging really a problem? Because we're a young country. But think about if we start now, only then we will be able to address the demands or the need of the young generation in future. And I was just talking to some of my students yesterday. Always in medical research, the young population is the guinea pig and the old scientists are testing on them. For once, the coin have flipped. The young population is going to test in the older guinea pigs what they can do so they do not get old the way we are getting old. So I think that's where the India needs to make a difference. So where is the gap? And you can see, unlike any other country, the trajectory of aging in India is very steep. We're going to get a lot of old people in India. And most interestingly, what we know in India is that almost 13 years of old age is actually spent in bad health status. It is much longer, much more severe than any other country in the world. And that means we really need to address it now so that the duration can be shortened. and the quality of life for the people can be ensured. And of course, for that there exists a big gap. We do not have a comprehensive multi-omic data set for the Indian. We need to have good quality data to understand what changes in Indian population when they get old, to understand what we need to do to develop interventions. All of you have heard about different clinical trials, biomarker discovery, studies happening globally. Now what happens in a biomarker discovery? You have a sick person and you have a healthy person. And you compare for signatures what emerges from a sick person which is not there in a healthy person. That's a simplistic viewpoint. But you have to think about that I need to get the signatures from a healthy person which is age, gender, socioeconomic background matched. Only then my signatures can be reliable. That means we need a good quality baseline data set. Now each study does this baseline data collection in their own way, their own fashion. What if we do not have to do that? What if we have a repository where the baseline data for India from various geographical locations, various socioeconomic background, genders, and different features are captured at a baseline level. So any future biomarker discovery can use this data as a reference data and test the validity of their biomarkers. That will be the value of such a baseline study, and that is what we are trying to do. So let's look at some of the studies which is done globally for multiomics, using multiomics platform to capture data for healthy population. And you can see there's a lot of nice blue circles you can see everywhere in the globe, which tells you that almost everywhere in the world people are trying to use multiomics tool for discovering new biomarkers. We are also there, somewhere a small blip, a dot right there. And the two studies also done in India, the Genome India, Phenome India, which is also there. But when you look at this graph once more with a different viewpoint and ask the question, how many of these studies are actually done by looking at aging as a parameter? Then you realize the number of dots are actually suddenly very few. And we happen to be the only one which are looking aging as a trajectory, as a parameter to collect healthy people data. There are a lot of other studies globally, and what is interesting is a lot of these studies actually initiated almost simultaneously of what we started. A-Bio study, aging biomarker in Singapore study, is something which was initiated almost at the same time we started our BHARAT study. So I think we are in the right space. We are doing something which is right, which is done globally at the same standard. Where we are making differences, we are collecting different type of data, which a lot of other studies are doing. And the challenge for us is something which was alluded by in the opening comments. The challenge is our diversity. Our diversity is so large that we can say that if you can find out features for aging for India, you can probably find features for the world, because the diversity is more in this country than in any given continent. So that is the challenge which we're going to fight, and I think this challenge is not going to be easy to address unless we put our forces together. So what is BHARAT study aiming to do? And this is what BHARAT study is about. We collect data from apparently healthy. I must say this is apparently because clinicians define if you're not sick, you're healthy. But that apparent health status is not which is easy to characterize. But we are trying to understand this by using apparently healthy individuals as volunteers who give their biological samples to us. And we collect these samples, hair, buccal swab, blood, urine, and stool, and we interrogate each of these samples using a lot of in-house and out tools which are available to the academic industry. We collect a lot of functional recordings, cognitive data, frailty assessment. We also do a lot of metadata, which means we collect a lot of information about who you are, what you're doing, and the way this whole thing is being done. And that's where the metadata is going to be very useful for curating what we're trying to do. What we capture are the details of metallions, hormones, and something very unique which we do, and Ramre is going to talk about that later, is the roughness of our immune cells. We use buccal swabs to look at glycation, a quick assessment of what the blood glucose levels are by using cheek swabs rather than a blood test. We do interrogation of blood, the vital fluid, because that's easily available material, by using multiple different tools, immune, hormones, blood biochemistry, and lots of omics. In urine, we do multiomics, and in stool, we do metagenomics. And I'll tell you in the next few slides what have we learned from these findings till now. And of course, when you have so much data, you need a lot of AI guys to come into picture, and we have a separate team of individuals. The chair of the session is one of that individual who will talk to you about using tools of AI. Now, this is a workflow. 1 year, that's what we did. And we stumbled, we fell. Except for bleeding, we pretty much did everything else. We realized convincing people, especially healthy people, to give samples is not easy. One, they do not come to hospitals. Why should they be? They are not sick. And if you're trying to find healthy people in hospital, there's something really wrong there. That either they are not healthy or they're not telling you the truth. So that becomes a challenge. So you need to go active recruitment for people. You do community level outreach. And that's where Carol from Baptist, who's here, she helped us reach out to the community at the level of the rural settings in Karnataka to sample healthy people. But once you figure out where you want to sample, you need to figure out what are you doing with the people? What kind of information do you want? While as a researcher, I knew what I wanted biologically. I knew this is a sample I want and I can put into my machines and get data. The data is meaningless unless we have good quality metadata. And that's just Professor Prabdeep, who's chair of ICPH, Isaac Center for Public Health, came into play. She actually helped us curate the metadata, the inclusion-exclusion criteria for picking these healthy people. And a lot of this information, which is done in the stage 2, once the volunteer is identified, we do on-site sampling. A lot of information was curated by Professor Prabdeep and their team. And we collect this data along with a lot of physical on-site examination and sampling. Now, there's a lot of dirty work on the ground. A lot of people who have to stand in the queue and have to keep this smile pasted on their face when they bleed the people. And when they start taking blood more than 20 ml, people start giving them eyebrows and say, you're going to bleed us dry. So we had to come up with very innovative measures to keep people interested. We had to put some funny masks on their face so they don't feel bad. And we had to give them the reason that because we bleed you and collect blood, your cortisol level shoots up, so we have to close your eyes. Well, honestly, the reason was we don't want to see them how much blood we were collecting. So it was a challenge, but we come around that, and it was a necessity which made us to do these things. And then the last stage is we take all these samples. A lot of them is processed on-site by our team, but a lot of them come to the labs where they are stored, biobanked, and cataloged, and then the samples are analyzed in bulk so that we don't have run-to-run variation, and we can collect high-quality multiomics data. Some observations. I must tell you, if you want to take pictures of these slides, they might change in the next few days, few months, or few years, because these are all early observations. Please do not try to draw any clinical conclusion or implementation diagnostic kits development based on the data which we show. Please do not take pictures. It might be beneficial for all of us. So what did we learn? We learned something interesting in a lot of other things. First of all, we are actually getting very few people age 75 and above who are apparently healthy. Challenge is not that they are not healthy. Some people are indeed healthy, but age 75 and above do not want a blood work to be done because what if I find something is wrong with me? They do not want to be part of a study or do not want to know if something is wrong with their health system. So that convincing them to give samples is a big challenge. Carol's team did a wonderful job, and we have some samples in this category. We are now actively recruiting senior citizens through JSS College, and we try to increase the number to get a good representation for our AI models to be developed. But what we got was, through our repeated trials, we managed to get equal gender distribution. And some of the distribution studies, which you can see here, this is the age distribution, underrepresentation of elderly population. What we also found that a lot of population was actually diabetic, which is not a surprise, but what was interesting is they had no clue they were diabetic. So when they walk into clinic, they are apparently healthy, nothing's wrong, you do the blood work and you find they are diabetic. And 30% of the population turned out to be diabetic. Now that's interesting, that means 30% of our sampling is not usable. And that challenge will persist, hopefully, in the same way in the few years, unless we change our approach dramatically. What we also found, which is well known, the hand grip goes down with aging. Hand grip is a test of muscle integrity or muscle composition. Not a surprise, but what was interesting is the hand grip strength of women was lower, and interestingly, the older women had a better hand grip than the men. So which is well known, the women age better than men. And not a surprise here, but this proves it. This is the undiagnosed burden of the diabetic population. Again, we are dealing with an unprecedented problem in Indian metabolic health. 30% Indians have undetected, undiagnosed diabetes. And if you put this information for urban Indian, the number tends to be higher. So which is a scary thought, but that's what the ground reality is. Let's look at the blood biochemistry and many different parameters. So we used different axes to find where we can segregate the population. The biggest difference in our features was coming because of gender. No surprise. Gender is the biggest discrimination of features which come from blood biochemistry. Second was diabetes, of course, glycemic index. And these graphs at the bottom shows what are the features which actually allow feature distinction between various population. Then setting, urban-rural divide. Urban-rural divide was actually coming as a two population. We still have a small subset for urban because we largely sample rural population, but it was a clear discrimination between blood features of rural and urban Indian. And of course, when you look at the age, which is what we were interested in, we did see differences emerging between the youngest of the young and the oldest of the old, especially in the features, which clinicians might say no surprise, because that's what we see all the time: apolipoprotein, lipid metabolism altered, vitamin B12, ferritin, and folate. We know, clinically, people know these features change with aging, but we have these features change with age. And if these features are changing in apparently healthy people, maybe we do not need to do anything to intervene about these features. We need to invest onto features which come in pathologically aged condition. So what next? I'm sorry, again. Now this is a story which Professor Ramre is working on. As I said, I'm going to talk about what each one of the investigator and team is doing. Now Ramre is always about bittersweet things. He always finds most complicated ways to address simpler problems. And he loves looking at things which nobody wants to look into. So he said, let me go to the field and see how many people are willing to open their mouth and give me a cheek swab. So he collects the cheek swab, and what he's asking, that he's asking a simple question. Environment, macroenvironment, my exposome, how does it interact with my microenvironment or my metabolism? And if this interaction can be captured by changes in the glycations or formation of what they call carbonyls or amadori products, which lead to formation of what we call as advanced glycated end product, which change the way most molecules in our body behave, can we capture them by cheek swabs? And lo and behold, what we find that we can. We can actually see differences in the population based on age in the cheek swabs by looking at the glycated end products, by looking at diabetic population, and also this feature correlates very well with the intensity which we collect with the skin age reader. Now let's move quickly. We also looked at immune profiling. Siddharth did that, and the finding was we see something which is globally known: CD4 population, monocyte population is enriched. But what we also found, the neutrophil population, non-mature neutrophil populations are also enriched. So probably our immune system is exhausted at a faster rate than what's globally known. This is epigenetics. A lot of epigenetics clocks are sold globally. We say, look at epigenetic clock, it will tell you what's happening. And what we found, there are lots of marks which are different in epigenetics for Indian population. But when you apply them to the known epigenetic clocks globally, we realize we're off by almost 10 years. So global epigenetic clocks don't work for India. We really need an Indian epigenetic clocks because almost 10,000 plus epigenetic marks are unique to Indian population, which means that our epigenetic clocks must be different from what we know globally. Now, some proteomic data. I'm not going to spend too much time because there is something which was generated just last week. We actually processed our serum or plasma into high-throughput proteomic platform. Thanks to Hano's group, the protocol was used by his advice, and we do see some differences between the young and age population. And this is something which we are doing, model which we're trying to develop to see what changes with age. This is called the topical learning, and we clearly see some features start to emerge in the elder population. This is something very unique which we're trying, and it is an ongoing exercise. And lastly, the gut biome, which is an ongoing saga. Ongoing saga, I say, because collecting stool from India is a challenge. Indians don't like to part with their stool at all. They're very, very protective about their stool samples. And getting them to give good quality stool sample was a challenge, so we had to actually go back to the drawing board. And Shantanu has been dealing with literally the shit problem for a very long time. And we hope in the coming years we'll have some solutions in our hand. At this stage, I just want to say where we are headed. We want to look at two specific regions. We have Gujarat state, who is willing to work with us on sampling another part of India's geography. We want to sample 2,500 individuals in each region, generate multi-omic data, identify key features, identify tissue-specific aging markers. This is what is the long-term intent. But of course, we need AI model. I'll stop here because I want my co-conspirators to come onto the stage and tell us exactly what things are working and what things are not working. So I request Carol, Prabdeep, Ramre, Shantanu, and Srimant to please come on the stage so we can spend some time. And of course, Siddharth. So we have got all my team here. Lots of people are still in the audience because they come into the play when we just start generating the data. So we're right in the very early stages of data generation. So I'll go in the order how we have faced or battled different aspects of Bharat study. So first question is for Prabdeep actually. Prabdeep, India has very unique geographical and cultural differences. We have specific challenges. And how do we ensure that we are collecting right quality, right type of data, and from the right type of individuals in this study?
Thank you. And hi, I'm Prabdeep. I'm a public health faculty at the newly established Tata at ISC Medical School. So public health is new to ISC, and we've been trying to figure out how to bring that perspective into our research here. So as we have had multiple discussion, I think fundamentally the way we do research in the world of epidemiology or public health is very different from the way research is done, for example, with biology research questions. And I think, as we have repeatedly discussed, one of the big challenges, how do you decide from whom the data should be collected? So when we look at it from a very traditional epidemiological angle, we actually like to look at representativeness of the population. But as you saw, the complexity in doing a study like this, where you are trying to get large amount of data, biomarkers, and so on, it is extremely challenging to actually get a representative sample. So I think that is the first issue. And in such situations, and I think you will hear more from my other colleague, but the main question is, okay, let's say even if you are able to sort of cover different geographic areas, once you have selected a place, how do you then pick individuals in a way that they kind of represent different age, gender, and different behaviors, and also more importantly, they are free of disease conditions. So one of the things which I feel we need to get better at is screening stage. And I think we learned that, as you showed, right, that many people actually don't know they have a disease. Yeah. So when we say healthy, and somebody says, I'm healthy, but what is healthy? Defining the healthy is challenging, and hence we need to think through our strategies on how we screen the population to pick different age groups. But I fundamentally also feel, Deepak, maybe we shouldn't be too sticky about it in some ways, because if large proportion of population has what we call as the risk factors, for example, little bit high sugar, little bit cholesterol, and so on, and if we leave them out, that is also problematic. So in a way, enabling a sampling strategy which represents population, which allows us to sample healthy individual, but also understand who are the other people who are forming large proportion of the population, like 15, 20, 30% people have these risk factors who may not have disease. And finally, collecting data. When we are collecting data, I think one of the biggest thing is how we balance how many questions you want to ask, yes, and how much blood you want to draw. At the same time, ensuring that, we have this thing like, if you keep on asking too many questions, people start giving random answers, and then the data is not very useful. So picking the good quality questions, ensuring that the test we do really useful, and from a fairly well-spread population, even if it's not strictly representative from a sampling perspective, it should have a geographic, age, gender, and other diversity represented. Yeah. Thank you.
So I think what we are doing with apparently healthy is okay.
Yeah, I mean, it's fine, but I think we can further dive deep into our sampling strategies and also see that we represent different risk factor groups, although they may not be healthy, but from aging question point of view, they are equally important, right?
We'll do that.
Yes.
So I think, Carol, the next point is to you. We have finished 1,000 samples with you, and we did a lot of them in the rural settings. What kind of challenges do you find in the field which we face? How did you get consent? And there's something which we get asked is how many were asked before we get 1,000?
Yes. So first, when you showed me your idea, it was very exciting. And I know the good quality data is important for whatever samples we were taking. And the protocol also, you wanted every biological sample a person can give, Deepak. So it was a big challenge for us to take. So we know that you need an ecosystem, and trust is the biggest enabler. So we have been working in communities for decades, serving for their illnesses and also improving the health. So we chose those particular villages. And what we did is, we looked into the questionnaire. The questionnaire takes minimum 1 hour. So what we did is, we cannot do it on a camp day. We made a system that people go to their communities. First, in the communities, we had multiple way of speaking to stakeholders, saying that what is our purpose, what's the process, and we will come, and also an ecosystem of care. If we identify anybody with any diseases which is not, we put a care system, subsidized pathway, so that we are taking care of their health also. After doing that, we had 2 to 3 researchers who went to homes. Per day, 10 each of them collected. So Monday to Thursday, they collected 30 to 40 full questionnaires. When we go to homes, they are not available, we went to the fields. We went to their workplaces. We went to schools, colleges, and took their timing, their convenient time, sat with them, had these conversations. So this questioning, what we found is, if it is a question, people don't like. But if somebody is so interested to go there and ask about their life, they were so happy to give that. So that was one of the most important thing. And on Friday, what we did is, we tagged all the barcodes, and we went to every house which given their data, given the sampler for both stool and urine. And the next day, Saturday, they're supposed to come for the camp. Camp starts at 8 o'clock. Before 9:30 is that cortisol. There is a smaller window. So we have to finish all the 40 to 60 during that time. So I think 10 to 15 from your side, from our side, 20, 30 people were there just before the sun comes out, and then sampled 60 to 70 people. So after that, it doesn't finish there. We take them back, dealing them, because there's also a whole process of giving this to another place without the identifiers. So after that, so this is a whole process I wanted to tell that it has to be done in a meticulous way. That is how this sample came, so the quality of the samples.
Largely what you're saying is based on trust.
Trust, yes.
Inroads. Okay,
Yes.
That's great.
Thank you.
Thank you, Carol. Ramri, quick question. You chose most unusual samples which typically are not collected. So what was the motivation, and what do you think this study aspect will tell us?
Hi, thanks, Deepak. Thanks for the question. So the idea was that most of the samples that we were collecting were in some sense invasive, or had certain social or cultural difficulties to come across and override. In that respect, as a once trained clinician, the idea was, how can we innovatively try to take something from inside the body that would not be unacceptable or difficult or painful to the individual, and still reveal something about the tissue state of the body. And therefore, the idea of cheek swab or buccal swabs came to us. we had to, of course, run sanity checks to understand whether the buccal swabs are reflective of the state of health inside the body, and which you very eloquently showed was an emergence of both macro-environmental as well as micro-environmental problems, if you will. And we were able to see that same kind of chemistry which one sees in HbA1c, which many of us must be measuring regularly, was also reflected in the buccal swabs. And so now, without even bleeding myself, I can actually get a state of the sugars which are catching my proteins and rendering them dysfunct. I just want to end by saying that, again, acknowledge the students who did this. So it was Anchita and Anvesha who have carried much of this hard work through. It's easy for us to, fancifully dream, but the ultimate implementation is down to the real students. Thank you.
Thank you, Ramri. So, Siddharth, quick question. It seems times have already elapsed for us, all thanks to the technical glitches in the beginning. So the immune system seems to show different features for India. So where do you think, what is probably different for India from your immunologist viewpoint?
Actually, a very good question. Thanks, Deepak. So what we've been seeing is that there are variations because of the environment. There's a larger proportion of tropical infectious diseases that everybody's exposed to. And this alters the immune microenvironment, especially in the circulating cells. That probably plays a part, but we're also seeing that there are innate immune cell differences, both in the monocytes and neutrophils in circulation, that are different than what has been reported by others in other cohorts. And so what the exact reason for this is something that we are not able to identify yet. We are observing a difference. That could be because of a potentially higher glucose level, epigenetic changes. They're all related, but how they're related is a question that we hope to answer over the next few years. Thanks.
Yeah, so clearly we need more data to make any claims first, right?
Absolutely.
Great.
Absolutely.
Great. Shrimanto, you've been looking at epigenetics throughout your life. And what do you think the epigenetic in India is different? Why should it be different?
I think that is quite obvious, Deepak, because when we started this project with that idea in the epigenetic context, because if you see our epigenome is highly sensitive to the environment. It has several advantages as well as disadvantages, context-specific manner. But if you look, it changes our epigenome very easily with the change of food habit, lifestyle, with the socioeconomic conditions, culture. So all these environmental factors really has huge sensitivity to our epigenome. It can be histone modification, DNA modification, anything. So that's why the clock made based on the Western population, what we see preliminarily, that it's not very well fitting with our DNA methylation data. And as you see, we can see a lot of unique signature for Indian population. But I'd like to just add, we have number of sample is very less as it is very sensitive to environment. So there will be a lot of confounding factor as well. So we need really a huge number of samples to really concrete way to establish clock and methylation signature for our population.
Yeah, thanks. Thank you. Shantanu, last question. I think before the organizing team starts giving me eyes. Our gut biome is influenced with what we eat. And in India, our diversity of food is so much that our gut genome or gut microbiome data will give us a lot of noise. How are we going to handle such a noisy environment?
Right. So thanks, Deepak. So the choice of words of noise here itself is something that we have to examine closely. We get a lot of variation in our sample, but I don't believe all of it is noise. There is genuine biological signal in this enormous variation that we're getting because the kind of sampling we are doing from such a diverse and heterogeneous population. But I do believe in this there is genuine noise, and that genuine noise has to be separated from this large extent of variation. This noise can come from multiple aspects, right? From sequencing to extraction to analysis to, there's a huge pipeline which is multiple parameters involved. our aim is now to separate this noise from this biological signal. So I think a lot of variation is expected, is good, is useful. But the good thing in our data set and our approach is that apart from the gut metagenome, we have this multiple dimensions of data from questionnaires, blood markers, lifestyle diets. We're going to quantify how much fiber they eat, how much protein they eat, how much stress they have, quote unquote. So these are going to be multiple clusters in data sets, and one cluster will be of the metagenome community. So now the challenge is to, how well these multiple clusters align with each other, and over and above that, what is the noise? So I think this is very challenging, but a lot of fun, and I think my group will really enjoy tackling these questions.
Thank you. I think we run out of time, but one theme which keeps emerging is we still do not know enough. We need more sample, more data, more information before we start making any claims for India. So at this point, I'll thank everyone, thank my colleagues for being part of this study, and I hope that we will join more people next year on this stage and we do the things. Thank you so much. Thank you, everyone.